So in today's lecture, we are gonna start with the NMG and sliding filament theory of muscle contraction. People feel this as a very difficult topic, but I'll try to make it very simple for you, and we'll discuss some of the important pathologies as well, which are related with your NMG and sliding filament theory, like botulinum toxin 1 and about the snare proteins. Most of the books skip the snare proteins, but snare proteins are very important for you to understand some of the basics of botulinum toxin, botulinum A, E, and all. Okay, we'll discuss slightly about that. And we'll start with the skeletal muscle, the structure of the skeletal muscle, and afterwards we'll go to the structure of the sarcomere, and we'll discuss about the events at the NMJ, at the neuromuscular junction. What are the events that are occurring in the body, and about the related physiology and pharmacology. We'll discuss this widespread chat. It is not difficult as it seems. It is very easy. Okay, we'll try to discuss it. We'll also, if time permits, we'll discuss the muscular dystrophies, the dysphenia, and the Becker's muscular dystrophy. And about the sliding filament theory, we'll summarize it in a flowchart manner, as well as we'll discuss it in written also in detail. Okay. So that is our agenda today. We have a lot to cover. It seems very less, but it's actually not. We have to start from the real beginning of muscle contraction, and as time progresses, we'll start discussing some of the intrinsic concepts which are there in your muscle contraction mechanism. Okay. So today's lecture is going to be very interesting. Stay tight, and let's start. So we are discussing the NMJ and sliding filament theory of muscle contraction today, basically. But the topic we are starting is the basic muscle fiber organization. We are discussing about the organization of a muscle fiber, how a muscle fiber is organized so as to facilitate the muscle contraction. Okay, so we are discussing about this. Let's start. So what you are seeing is a skeletal muscle. Okay, in the skeletal muscle, when we are talking about a skeletal muscle, we are not talking about cardiac muscle now, because cardiac muscle, the muscle contraction in the cardiac muscle is slightly different from it from a skeletal muscle point of view. So we are taking our prototype as the skeletal muscle and discussing the things. So if this is a skeletal muscle, the outermost covering of the skeletal muscle is known as the epimysium. Okay, so the epimysium, what is it? Just mark my words very carefully. It is the outermost covering of the skeletal muscle, outermost covering of the entire skeletal muscle. Okay, and this epimysium is basically a dense, fibrous, irregular connective tissue. Okay, so this epimysium is a dense, fibrous, irregular connective tissue in nature. Okay, this muscle fiber, when you are not muscle fiber, the skeletal muscle, in fact, the skeletal muscle as a whole will consist of groups of some groups. Okay, just to understand, some groups, and this group is a single group is called as a fascicle. Okay, so a skeletal muscle is in fact a collection of fascicles. Okay, so muscle is a collection of fascicles. So what it is extrapolated here, this one, this pink arrowed one, is the fascicle. Okay, this is a fascicle that is brought outside so as to visualize you the structure. Understood? So muscle fascicles are there. You can see many groups of muscle fascicles in a skeletal muscle. Okay, understood. So this muscle fascicle, the muscle fascicle will be covered again, and this covering of the muscle fascicle is known as the perimysium. Okay, and this is also dense, fibrous, irregular connective tissue in nature. Okay, the outermost covering of the muscle fascicle is called as the perimysium, and this perimysium also is a dense, fibrous, irregular connective tissue. Okay, so if you understood this much, let's move on and look at a fascicle in individuality. So this is a fascicle. This is a fascicle. And this fascicle, if you can see, the outermost covering is of course the perimysium. Outermost covering is the perimysium, and each fascicle is a collection of muscle fibers. Is a collection of muscle fibers. So fascicle is a collection of muscle fibers. Okay, fascicle is a collection of muscle fibers. So here each muscle fiber is being extrapolated out. This is a muscle fiber, and the outermost covering of each muscle fiber, outermost covering of each muscle fiber is called as your endomysium. So this covering, this whole covering of a muscle fiber is known as the endomysium. Okay, this is known as the endomysium. Understood? Okay, so endomysium is the covering of a muscle fiber. Covering of a muscle fiber. Done. You understood what is epimysium, what is perimysium, what is endomysium? Okay. So this is very integral for you to understand this. And if we take out a muscle fiber, in fact, in totality, okay. So here we took out a muscle fiber, and we are seeing now a single muscle fiber here. Okay, this one here. Just visualize. Yeah. Okay. So now here we are seeing the muscle fiber. Look at this. It is not this one as it is marked. This is not this part. It is the whole part which we are seeing now. Okay, the muscle fiber is shown here. Okay, just don't be confused. There is just a simple glitch in this diagram. Okay, the muscle fiber is just zoom out here, and this muscle fiber, if you see, is a collection. This muscle fibers, in fact, is a collection of myofibrils. Myofibrils. It is a collection of myofibrils. This muscle fiber, uh, don't get confused. This muscle fiber is known as the muscle cell. This muscle cell or muscle fiber is also known as the myocyte. The three are the same terms. Okay, don't get confused in the terms. Muscle fiber, muscle cell, or myocyte. This is the muscle fiber, muscle cell, or your myocyte. Okay, and this is the myofibril. And this myofibril will have the myofilaments. This muscle fibril will have the myofilaments, that is the actin and myosin. Okay, and actin and myosin will be arranged in this particular thing. Okay, if you want to see a myofibril, this is a myofibril. Okay, this whole structure, and you can see the actin, the thick filaments or the myosin filaments here in the red. These are your myosin filaments. We'll see how they are arranged, and these are your actin filaments. And these are your actin. Okay, and here is a line, a zigzag line. So zigzag Z. Okay, so start from Z line. Okay, a Z line. We'll understand what is Z line and why it is named so. Okay, it is a German term, Zissen-CB. Okay, so it's a Zissen-CB line, and a M line is there also. Okay, H zone is there. We'll see how they are organized. Just understand the distance, the part of a muscle, the part of a myofilament. Okay, you can say a myofibril or myofilament. The part between two successive or two consecutive Z lines. Between two consecutive Z lines is called as a sarcomere, and sarcomere is the functional unit of contraction. The sarcomere is the functional unit of contraction. Why it is called so, we'll see. Understood? So if you got confused in the above part, which I feel so you must have, because there is a technical glitch in this diagram. The This one is marked muscle fibers, but uh, what is it extrapolated here is marked muscle fiber, but it is not. This muscle fiber is drawn in the same fashion below also. Okay, no problem. I will draw another hyperschematic diagram for you. Let's assume this is the muscle. We are taking a cross section. Okay, this is the muscle. If this is a muscle, the surrounding structure of the muscle, what it is called? The surrounding structure is the epimysium, right? So this one, let's assume epimysium. So now this is the muscle. This is the muscle. Okay. So muscle, we told, is a collection of fascicles. So let's draw the fascicles. There is 1, 2, 3, 4, 5. All are not circular in nature. Okay. Variable shaped fascicles are there. So no need to point out mistakes. Yeah. So these are your fascicles. This fascicles, just a minute, a call is there. Yeah, sir, back. Yeah. I was telling that this is your epimysium. These are your fascicles which I have drawn. And here are the fascicles. If these are the fascicles, the surrounding structures of each fascicle, this pink one will be your perimysium, right? Perimysium. Yeah. And this is a fascicle. This is a fascicle. This is a fascicle. Fascicle. Just a similar sounding term is there. I'm telling you, it is fascia. Okay. After fascicle, there is such a similar sounding term. People get confused between fascia and fascicle. So fascia is nothing but the connective tissue between the fascicles. Like this space, in this space, in this space, we have some connective tissue present, and this connective tissue is called as a fascia. So fascia is nothing but the connective tissue between the fascicles. Okay. So no need to confuse it with fascicles in any manner. Just forget fascia. Okay. It's not of much significance in our discussion. Okay. Again, this fascicles will be the collection of muscle fibers. Muscle fiber, muscle fiber, muscle fiber, muscle fiber, muscle fiber. Done. These are your muscle fibers. There must be covering up the muscle fiber itself. What is this called? It's called as your endomysium. This called as your endomysium. Understood? So endomysium is the covering of the muscle fiber. Okay. So if you understood this, let's move ahead. And what I said is a muscle fiber. A muscle fiber is made up of your myofibrils. So there must be myofibrils inside them as well. Let me take, yeah. So if this is a muscle fiber, there must be small myofibrils here. These are, what are these? These are your myofibrils. Okay, these are your myofibrils. And these myofibrils, uh, if you see a structure of myofibril, you will get to see such a structure. This is the structure of a myofibril. Understood? And this myofibril will have your sarcomeres present. Uh, yeah. So these are your myofilaments. Myofilaments are sometimes used synonymously, but actually myofibrils contain these myofilaments, and these myofilaments are basically nothing but your actin and myosin and some associated proteins as well. Okay. Yeah. So endomysium and sarcolemma, two things people confuse much. Okay. Which I'm discussing, just see. Two things are there here. One is endomysium, one is sarcolemma. So if we'll see a muscle fiber, Let's assume this is a muscle fiber. So the immediate surrounding, the immediate covering that is surrounding the muscle fiber is your sarcolemma. Okay, I'm drawing it with red. This one, the immediate one, is called as your sarcolemma, and the covering that is outside the sarcolemma. Okay, the covering that is outside the sarcolemma is called as your endomysium. This purple one is your endomysium. Okay, sarcolemma is basically the plasma lemma of the cell, and endomysium is basically the covering. So if it is a cell, the immediate surrounding must be plasma membrane itself. Otherwise, it is not a cell, right? So sarcolemma is basically the plasma membrane, the cell membrane of the muscle cell or your muscle fiber or your myocyte, whatever you can say. Understood? So if this is clear, okay, if you understood this all facts, if you understood this whole diagram, the cross section, the extrapolation, as well as the up to sarcomere, if it is clear, then we'll move to the sarcomere. And once we discuss the sarcomere, it all becomes very easy for us to understand the muscle contraction. Then we'll move to the NMJ directly. Okay. So we are discussing now the sarcomere. Now discussing the sarcomere. Okay. Okay. So sarcomere, if we are discussing the sarcomere here, although this is a diagram here, it is better if I draw it for you, right? Okay. I believe that we should read in the chalk and talk method itself. Just events at NMJ, will discuss it later. Just a minute. Let me sort the pages. Let's add a page and understand. Yeah. Come to here. Let's read about a sarcomere. If we are discussing about a sarcomere, we have to see A band and I band first. Okay, A band is thicker and it is made up of a myosin, and it is called A band is called as a A band because it is anisotropic. That is, if you look at electron microscopic picture of the A band, it appears very thicker. Okay, it appears very thicker. That is, it is more refractile. If it is anisotropic, one thing is called isotropic and anisotropic. if if or something, okay, just assume something has irregularities. You must have read about anisotropism and isotropism in a solid state chapter in 12th, yes. Yeah. So anisotropic, so if we look at the electron density, it is irregular, so it is anisotropic. It is more refractile than the isotropic part, okay, or the I band part. So anisotropic is there. So A band and I band. We will draw the A band as well as the I band for us to complete the sarcomere. So let's draw first the A band and the I band. Look at this. These are your A bands. Assuming these are your A bands, these are thicker and anisotropic. And we'll draw the I bands as well in the parallel fashion. A bit thinner bands are the I bands. These are your I bands. Okay. Let's draw more. Done. So A bands and I bands are there. And then in the middle we have a M line present. Okay, we have a M line present. So here is your M line. This is your M line. Okay, and again we have, just look at this. These are the I bands of the neighboring sarcomere. We'll connect this, how they are connected we'll see. These are the I bands of the next sarcomere that will form this. Okay, so there is a pattern that is going on basically, and there will be a Z line. Z line means a zigzag line that will be connecting the I bands in a zigzag fashion. This one, this blue one, is basically your Z line. And this Z, it stands for Zishan Sibi line. Zishan Sibi line. And this Zishan Sibi is a German word. It is a German word. This M in the M line stands for Mittelline, that is a midline. Okay, this is also a German term. Okay, and this Z line is often called the Z disc as well. So here we have a Z line or the Z disc present. Here we have a M line present. These black ones are the I bands. This red ones are the E band. Okay. Now we're wondering how the A bands are connected like this, no support and nothing, how they are present. Okay. So there are some proteins which are present, spring-like proteins, which link the thick filament, will not going to thick filament, which link the A band to the Z line. Okay. So here let's draw it in green. Yeah, look at this. I have not drawn the Z line here. I'll draw it. Let me draw the proteins first. Okay, and the Z line of the opposite fragment as well. Mm, yeah, done. So what is this green protein called? This green proteins are what called as the titin protein. The titin protein. And the titin protein, the function of the titin protein is to link the A band or the thick filament to Z line. So basically, it is a supporting protein. There is a titin protein or the supporting protein. Okay, so now we will define A band and I band. Okay, now we'll define A band, I band, and the H zone. What is the H zone? Let's first come to the H zone. So, if you can see, there is a zone here, the middle zone, which is free from all the I filaments. This middle zone is free from all the I filaments. So, the zone which is free from all the I filaments, this one is called as your H zone. And H here stands for the Hansen's zone. Okay, the Hansen's zone. H zone or the Hansen's zone. Understood? Okay. Now we'll look what is the A band. Okay, A band. Just focus here. Starting from here and here, this part. This part is called as your A band. This is your A band. So A band, what will A band contain? Okay, we'll discuss. A stand for anisotropic band. That is, it is more refractile, which means that it is more electron dense on electron microscopy. More electron dense on electron microscopy. If time permits, I will show you an electron microscope picture. Just give me a minute. I will bring a picture here. Yes, so now you can appreciate. Look at this. This picture is the electron microscopic description of a sarcomere. Okay, and if you're seeing the electron microscopic description of a sarcomere, you can see here there are some dark areas and some lighter areas as well. Okay. The dark areas are the areas where there is more electron density, that is, more anisotropism is present. So here we have the, look at this, the A band plus your I band. Here also A band plus your I band. Do you know which area it is corresponding to? This part, this part, where there is A band and the I band intact. And this whole part, this electron density is the M line present. This pale part will have the A band only. This is the Z line. This has only I bands present. It has also only I bands. This is again the Z line. Okay. So more things are there, more electron density. I band is present, more electron density. Basically, this is the concept, okay, of a electron density that is there. If you understood this, let's now define the I band. What is the I band? Starting from this part, from where the A band ends and to the next point where the A band will start. Here, let's say A band starts next. It will continue, right, like this. So this part, this portion, the purple portion is called as your I band. Okay. Question number 1. A band has myosin or actin present. Just a minute, there's another call. Yeah. Hello? Yeah. So we are talking about your A band and I band. I hope you completed the A band and I band. Yeah. I band is called so because it is your isotropic band. I got distracted from where I left over because there was a call. Okay. This I band is your isotropic band. That is, it is your less reflectile. Okay. Isotropic band means it is less reflectile or less electron dense on electron microscopy. And we saw the electron microscopic picture of this also. Okay, so here we completed the actin and myosin, and let's look at the diagram. We discussed all this, right? I band, A band, yeah, all are done. So A band, I told, let's look at this part. I band, this whole part is I band. This is your I band. This spring-like protein is called as your titin. This is your H zone, which is free from all I bands. This is the thick filament, which is the myosin, and the thin filament, that is your actin. Purple one is the actin, thin filament, and this red one is the thick filament, that is myosin. Just a minute. Yeah. I band. Okay, understood. So there is a particular arrangement of actin and myosin. If we look at the transverse section, then you will see that one myosin is surrounded by 6 actins. One myosin. This purple one is myosin, right? No, this red one is myosin. Yeah. So I'm drawing it with red only, and actin I will draw it with black. Okay. So one myosin. Let's say this is myosin, this is myosin, this is myosin. One myosin is surrounded by 6 actins. So we'll draw 6 actins here. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, and 6. Okay. And one actin is surrounded by 3 myosins. One actin is surrounded by 3 myosins. So it is evident here. Let's say this is one actin. This purple one is surrounded by three myosins here. And this one myosin is surrounded by six actins here. Can you see the pattern? Okay. So here we write one myosin surrounded by six actins, and one actin is surrounded by three myosin. Okay. So if this is clear to you, if this part is clear to you, let's move ahead and start discussing about another thing, that is we'll start with actin and myosin is done. We'll see how a F actin is forming the troponin and tropomyosin and all those concepts. Okay, so just focus here. Just focus here. What I'm saying. We discussed about actin and myosin, but there are more proteins that are used in the muscle contraction. Okay, so basically, when we are taking a monomer, we'll take a monomer G actin. Okay, so one circular thing, right? This one, this is a heart. Okay, this is a circular thing. So this circular thing will repeat and repeat to form a filamentous thing like the stylus, and this filamentous thing will be the F actin. Okay, this will be the F actin, and this filamentous thing will have a like rope winding around like this. This is the tropomyosin. A rope like protein is the tropomyosin. Okay, so this is our orientation. We'll start with G actin. So actin is two forms, G actin and F actin. G stands for globular, F stands for filamentous. So this is a G actin or globular actin, which is the monomeric unit. And when this monomer repeats itself in the form of a filament, it is called the F actin. And F here stands for the filamentous actin. And this is the polymer. This F actin will have a rope like protein that will be blocking the active actin binding sites on it on the resting stage. If a muscle is not contracting, the actin will have one site which is your actin binding site. Okay, just to understand, active sites on the myosin binding sites. We'll discuss about that. Don't get confused now. Just look at this. A rope like protein will be there, like winding around like this. This is the tropomyosin. The tropomyosin. Okay, tropomyosin is not TROPHO, it's TROPO. Okay, tropomyosin. Understood? And this tropomyosin is blocking the myosin binding sites on actin. What happens in muscle contraction is that on actin there are actin myosin binding sites. Okay, so myosin head binds to the myosin binding sites upon the actin. Okay, and pulls it, and muscle contraction occurs. This is a basic introduction. But what if the tropomyosin is wound around like this? So it is blocking the myosin binding sites which are present upon the actin, and if it blocks the myosin binding sites present upon the actin, then there will be no attachment. There will be no cross bridge formation. It is called as a cross bridge formation. So there will be no cross bridge formation, and if there is no cross bridge, how it will contract? Okay, there is no muscle contraction. So in resting stage, when we have no muscle contraction, the tropomyosin is tightly wound around, covering the active sites. Okay, so this tropomyosin, what it is doing, it is covering the active sites for binding of myosin head on actin. Understood? And if you want to look at the structure of a myosin head, okay, there are two like parts, like a heavy meromyosin and light meromyosin. Not much of clinical significance, just the molecular weight difference is there. Just assume this is one, and this is its head. Okay, this is a snake-like structure. Okay, this is not myosin. This is a meromyosin, the monomer of myosin, and this many monomers will continue to form the myosin. And like this, it will have another head like this. Okay, two heads, and here is a neck. This is a neck region. Okay, this is the structure of a meromyosin, which is the monomer of myosin. Is the monomer of myosin. Okay, and this part is the tail. It has two heads. Okay, like the hood of two snakes. Okay, there's two head, and here a important enzyme is present. Just remember it forever. It's called as a myosin. Okay, myosin ATPase enzyme. Myosin ATPase enzyme. We'll see its significance soon. ATPase means what it will do. What will be the basic function of this enzyme? It will break down the ATP into ADP plus inorganic phosphate. Okay, it is the work of the myosin ATPase enzyme. Okay, and another site will be there, that is the actin binding site. Two sites will be there. This is the actin binding site. I am writing ABS. This actin binding sites will bind to the active sites on the actin filament. Understood that? Okay, so now what we are going to see is okay, more to discuss. We haven't discussed about a protein which is called the nebulin protein. We'll discuss the nebulin protein first. Let's assume there are two effectins which are wound in this fashion. This is one effectin. This is another effectin. I know drawing takes a lot of time, but the concept that is impacted on your heads is just amazing. It will stay for long. That's why I'm trying to draw everything that is I'm telling. Okay, and there is a backbone which upon which these effectants are wound. This backbone is formed by a specific protein which is called as a nebulin protein. This is your nebulin. And of course, tropomyosin will be there, right? Tropomyosin will be like rope like. It will be like this, somewhat like this, blocking all the active sites on the effectant. Will block all the active sites. Yes. Upon this tropomyosin also, there is a protein which is called as a troponin. Okay, troponin, like this. This is your troponin protein. We'll draw like this. This is your troponin protein and has a very, very important role in the muscle contraction. We'll see. So this is your troponin. Troponin. Troponin is very important diagnostic point of view also. Troponin is the earliest marker to rise in your myocardial infarction. Okay, so everything we are reading is basically linked to the clinical subjects. So don't think it as a waste. Troponin is a cardiac biomarker. If you have read in biochemistry, and subsequently in medicine lectures, you'll read troponin. So troponin will have three parts. Okay, it has three parts, three portions: TPI, TPT, and TPC. So if I discuss just a schematic structure, this is a schematic structure. So this part of it will be called as a troponin I, TPI, that is inhibitory, TPI, and here I stands for inhibitory. This part will be called as a troponin T, troponin T. Okay, and this cleave-like part will be binding to an important element, that is a calcium, and initiate the muscle contraction. This is called as a troponin C. C stands for your calcium. Okay, so three parts: troponin I, troponin T, and troponin C. Understood? This is the basic thing which is there for a which are the base for your understanding of your muscle contraction. So I hope you understood the sarcomere, you understood the electron microscopy diagram, you understood the structure of troponin, structure of your F actin and G actin, how they're coiled. That is the basic structure of the actin. You understood the basic structure of the myosin. You understood what is troponin, what is tropomyosin, and nebulin, titin, etc. Yeah, done. Okay. Another thing is that actin is linked to other proteins of the sarcolemma as well. Okay, this actin, this actin is linked to other proteins of the sarcolemma as well. And the main thing, and the main protein that links actin to other proteins of the sarcolemma is called as a important protein, which is dystrophin. Dystrophin. You must have heard that the dystrophin gene is the largest gene in the human body. Okay, and the important mutation, the mutation in this dystrophin, if there's a mutation in dystrophin, we call it the muscular dystrophy. Muscular dystrophy, and it's an X-linked recessive condition. Okay, X-linked recessive condition. And muscular dystrophy, if we talk about muscular dystrophy, there are basically two major types. Okay, one is called the DMD, that is a Duchenne muscular dystrophy, and one is called the BMD. One is the Duchenne muscular dystrophy. One is the Becker muscular dystrophy. Two muscular dystrophies are there also. This DMD and BMD, not much is needed at your level. This DMD is basically a more severe condition. Okay, it is more severe than your Becker's muscular dystrophy. Becker's is a bit less severe. Basically, it's due to a missense mutation. Missense mutation, when the misfolded protein is formed, and basically all things are happening. We'll discuss in future someday about the differences and in detail about the muscular dystrophies. Now it is not much of clinical significance. So the main goal of our class is the events at the NMJ, the neuromuscular junction, and how the synaptic transmission and all things are happening. Okay, you understood all this? Yeah. So I know a lot of time is taking, but just hold on. We'll reach to a fantastic conclusion of muscle contraction. Okay. Let's discuss about the events of the NMJ. What are the events that are occurring in the neuromuscular junction? We'll see how the neuromuscular junction, what is happening and all. About basics of a neuron. Neuron has a synaptic end terminal, the synaptic end plate like this, synaptic knob. Okay, and many things are occurring in the synaptic knob, and basically the neurotransmitter is being released, and this neurotransmitter will go on to act on the the motor end plate part and depolarize the L tubules, the T tubules, sorry, this T tubules, and this T tubules will act on a receptor, the rhinodine and DHFR receptors. Okay, and this will push out the calcium out of the sarcoplasmic reticulum. This calcium, which is pushed out of the sarcoplasmic reticulum, will basically bind to the TPC part, and if it binds to the TPC part, then the tropomyosin will unwound. If the tropomyosin is unwound, there will be formation of crosslinks, and if there's formation of crosslinks, there will be a power stroke here, and the power stroke, there will be muscle contraction. The muscle contracts at the molecular level, the myofibril level, and this myofibril level gradually, not gradually, myofibril level will be at a microscopic level, and when it appears on a macroscopic level, we see that your biceps is basically contracting. Okay, there's a contraction of the biceps. This much things are happening until we are able to contract a muscle. Okay, so that is the molecular mechanism, the beauty of medicine. Okay, that you are able to understand the microscopic details of what we are seeing by our naked eyes. Yes. Okay, so now we'll discuss the events at the neuromuscular junction. Not neuromuscular junction, will start straight away from our mm, spinal cord itself. Okay, so all the impulses, this somatic motor neurons is somatic activity. So somatic activity, it originates from the anterior ventral gray horn. Okay, anterior or ventral gray horn. Understood? So impulse came here, impulse came here, and the nerve, this the whole course of the nerve neuron. Okay, it comes via the alpha motor neuron. Okay, so basically talking about alpha motor neuron, because somatic response is there, due to which you have to contract the muscle, and this somatic muscle, this impulse will be generated at the anterior horn of spinal cord. This will come by alpha motor neuron. Which neuron? Alpha motor neuron. There is another gamma motor neuron. Gamma motor neuron will innervate other things like this is separate concept of a nuclear chain fibers, bag fibers, okay, and extrafusal fibers, intrafusal fibers. How they are stimulating one A and all. It's a separate concept. Just leave it. Basically, the alpha motor neuron is here in our muscle contraction mechanism, which we are going to discuss. So impulse came via the alpha motor neuron, came down. Okay. So neuron, it has like this. It has a axon and some myelinations and a synaptic end plate. So impulse came down here and reached the synaptic knob, the junction, the trigger zone. Trigger zone is present and the synaptic knob is reached the trigger zone, and this impulse will facilitate the opening of the sodium channels, which will cause the sodium inflow, and the basic action potential will generate. The basic action potential will generate. So what will be happening in the synaptic knob, we'll see via this diagram. Basically, I'm drawing one. Just pay very minute attention to what I am drawing over here. Okay. Impulse reached here. It came by this. Okay. And when it reached the sodium channels, here we'll draw some sodium channels. Let's try it with green. I'm zooming it in. These are your sodium channels. So when the impulse reach the sodium channels, they will stimulate the sodium channels, and whenever impulse reach, we know the depolarization will occur. Okay, so there will be sodium influx. So sodium from the outside environment will inflow into the muscle like this. There will be sodium influx and creation of depolarizing wave. Okay, whenever there is sodium influx, there is lots of positive charge we are gaining. This positive charge will cause a traveling of a depolarizing wave, which I am writing with dw. Okay, understood? When this positivity reached a threshold level of plus 30 millivolt, this part, this part, I believe. In nerve muscle physiology, this is the basic part which you need to know. In action potential reaches the plus 30 millivolt. Okay. Its second thing will be happening, that is the influx of sodium, influx of calcium into the cell. So here we are gonna draw beautiful calcium channels. These are your calcium channels. So when it reaches plus 30 millivolt of potential, there will be calcium, which will be influx. There will be influx of calcium into the cell. That is, now we are gaining lots of calcium as well. Okay, easy. Okay, these two things are happening. We gained calcium, we gained sodium. So basically, now this calcium is of our importance. Okay, understood? So what is happening parallelly, that is sodium ion diffusion to synaptic now, calcium ions inflow into the cell, okay, and then there will be some proteins that will be acting over here. Okay, just very carefully look at my diagram now. There will be some vesicles, and this vesicle will contain what you already know is the ACH, that is acetylcholine. Acetylcholine. How we are making the acetylcholine? We'll understand this also. In one diagram, we'll finish all of the physiology here. Okay. So basically, there's a transporter via diet. Okay, via your diet, you are getting choline. Choline you are getting via your diet, and acetyl group you are getting from your COA. Okay, via choline you are uh, getting this choline inside. This choline is coming inside. This choline came inside. Okay, and here in this choline, by this choline transporter, of course, I'm writing CT. And this choline, there is an enzyme which is called as a choline acetotransferase, CAT enzyme. Choline acetotransferase, in which the COA is released out, and this choline and this acetyl from this acetyl COA, basically the COA is released out, and only the acetyl group is there. So this acetyl group binds with this choline. Just a minute. And this is forming your acetylcholine. This is forming your acetylcholine. Okay. So acetylcholine is being formed over here. That is choline. From choline we are getting, and this choline is basically acted upon by enzyme CAT. That is CAT is choline acetotransferase, and this choline acetotransferase is uh, removing the acetyl and COA, is breaking the bond between acetyl and COA, acetyl group and COA, and this is adding the acetyl group to the ACH. Okay, so basically this is the whole fact. This is the whole fact of what it is having, and how it is going in. This acetylcholine is acting via a transporter here in the vesicle itself. If you want to know, this is a vesicle, right? There is a transporter, ACH H plus transporter. There is a, here is the transporter ACH H plus, okay, ACH H plus antiporter. By this ACH H plus antiporter, this acetylcholine, I'm writing ACH from now, ACH is moving inside, ACH is moving inside, and what will be coming outside, as it is an antiporter, this H plus ion will be coming outside. And this H plus in the this part, okay, this H plus is regenerated again. This H plus is not lost here from here. This H plus is regenerated again by using your ATP. So this H plus again is regenerated from H plus from outside, comes over here, and this H plus, as it is moving against the like concentration gradient, it will be using the energy from the ATP itself. ATP, and ATP is being hydrolyzed to ADP plus PI. So we are needing ATP so as to bring the ACH into inside. So who provides ATP? Mitochondria. So we'll have tiny beautiful little mitochondria that would be providing this ATP. Did you understand the whole physiology? How is ACH is being formed? It is being transported inside the vesicle and all. Did you understand? If you understand this whole physiology here, okay, it is very easy for us to extrapolate more. We saw we had little calcium here. Okay, we had calcium. So basically, this vesicles will have a B snare and a T snare. Okay, so basically we have two type of snare proteins. There are snare proteins. Snare proteins are basically fusing proteins. Snare proteins. So snare proteins we have two types: T snares and V snares. T snares and V snares. So T snare and V snare. An important example of T snare is a synaptotagmin. Okay, synaptotagmin. And an important example of T snare is SNAP25. These two proteins will be fusing with each other. T snares and V snares always fuse with each other. Okay, another examples are there. An important example again of V snare is synaptobrevin. Synaptobrevin. And an important example of T snare is syntaxin. And these two will be fusing with each other. Done. How they are present and what is the thing? Okay. So T snare, always remember, they are present at the postsynaptic membrane. So what is the presynaptic and postsynaptic membrane? You got to understand. If you don't know, this is a synapse. This is the presynaptic membrane. This is the synaptic cleft or synaptic gutter. This is the postsynaptic membrane. This is a vesicle. So always remember, these T snares are present over here. These are your T snares. And this V snares are present over here. These are your V snares. An important analogy. Let's say you have a very special friend with you who lives far from your dwelling place or your home or your hostel or somewhat. Okay, and there comes a third person which will try to surprise you both by arranging a secret get together. Okay, like that. So basically, this T snares and V snares are far from each other, and there must be a connecting link between T snares and V snares so that these vesicles can fuse to the cell membrane and release the acetylcholine out. Okay, so this linking part is your calcium. Now, can you fit what will the calcium do which came inside? This is the connecting part. This is the connecting link. So basically, this is the connecting link between the V snares and the T snares, and this will help the proteins to fuse. This vesicles will fuse. Exocytose the ACH. This ACH will come and bind to the postsynaptic ACH receptors, that is the nicotinic receptors. You know about nicotinic and muscarinic receptors? These are your nicotinic receptors for ACH, which are the ligand gated receptors. The ligand gated receptors. Okay. So basically what I said is that we have our pretty ACH here. Here will be a snare protein and here will be another snare protein. Let's say in the same, okay, another snare protein here and another snare protein here. Okay. So T snare will have your SNAP25 here and we'll have a syntaxin here, which will be fusing with synaptobrevin and with your synaptotegmin. And what will help in this fusion? This pretty calcium will help in their fusion, and when they fuse with the help of this calcium, so they fused here, they fused like this. And when they fuse, they will cause exocytosis. They'll cause First they'll cause fusing. Okay, one is called the priming. Okay, priming is the process in which the vesicles get ready to release their content, and another thing is called the docking. Okay, so let's say here is a vesicle. Okay, I'm using this heart more often, no problem. And here is our postsynaptic membrane. So priming is the process in which these vesicles get ready to release the ACH neurotransmitter or contents out. Okay, this is priming. And when it gets fixed, docked, dock, DOCK, yes, this is docking. So after there is this phase of priming and docking, is not much of our importance. It releases the content out by the exocytosis. And content, what is the content? ACH. So this fusion, after this fusion, there will be a process of priming, there will be a process of docking, and a release of contents out. There will be release of contents out, and this contents will be ACH only, and this ACH is a neurotransmitter, so it will bind to nicotinic receptors, and after binding there will be a action potential in your post synaptic membrane, and our impulse gets traveled. Understood? So here is one. Okay, we could discuss a little less, and I am running out of time. No problem, guys. We'll continue this. Okay, let's discuss here some of the physiology up to what we discussed. Okay, basically, we discussed the muscle fiber structure. We discussed the sarcomere. The structure of muscle fiber is done. We discussed the synaptic transmission. Muscle fiber structure and synaptic transmission are discussed. In the next class, we'll be discussing the neuromuscular junction, events at the neuromuscular junction, and the actual muscle contraction. So two videos are just like a web series. You have to watch the two videos in complete sequence. Understood? But we are not done with our class yet. We'll discuss some of the pathologies here. Okay. One pathology I told you, that was the muscular dystrophy. Another pathology, you know well, there are autoantibodies against ACH receptors. Autoantibodies are generated against the ACH receptors, and this autoantibodies uh, prevent this ACH to bind to this receptor by binding to them. Okay, they block them and prevents the muscle contraction as a whole. And this disease is called as a myasthenia gravis. You know this severe autoimmune degenerative disease of the muscle. Another thing is that Lambert-Eaton myasthenic syndrome, often called as a LEMS, L-E-M-S. What is that? Here we'll get autoantibodies against the calcium channels preventing the ACH release. Okay, now look at this. So here in LEMS is basically, it is an autoimmune disorder as well. And here autoantibodies against your calcium receptors are there. Calcium receptors. So there will be no influx of calcium. There will be no calcium influx, and if there is no calcium influx, there will be no fusing of the snare proteins. And if snare proteins aren't fused, ACH will not be released, right? This is LEMS. Understood? And what are the symptoms? Weakness improves with activity, dry mouth, impotence, and constipation. These are all related with your muscle contraction as well. Okay. Next, we talk about yeah. So voltage gated calcium channel. Then second step is calcium binds to ACH vesicles, triggering snare complex formation and fusion with the presynaptic membrane. I hope you can now imagine what I am talking about. Yes, very clearly. And if we aren't still done with this part, just watch this part because it is a bit molecular level, so it may be a bit complicated for you to understand at this level. So please do watch. It is important, in fact. So this botulinum toxin is basically causing the cleavage of snare proteins, which will prevent the release of ACH. So botulinum toxin, okay, the botulinum toxin is basically causing the cleavage of snare proteins. It is breaking down the snare proteins, and if the snare proteins are in there, there will be no release of ACH. The ACH won't be released. And if there is no release of ACH, there will be flaccid paralysis. There will be flaccid paralysis because there is no muscle contraction. The muscles will be flaccid. There will be flaccid paralysis, not spastic paralysis. And cranial nerve 3 paralysis and decreased reflexes. Decreased reflexes basically is called as a hyporeflexia. So hyporeflexia is an important feature of this botulinum toxin induced paralysis. Okay, and remember this botulinum toxin is often used by this celebrities to look prevent aging. So Botox is the trade name. Okay, Botox is the trade name of this basic. It is a type of injection. Okay, type of injection in which is taken over the face so as to look younger. Okay, it is basically used by your celebrities. Okay, so as to look younger also. So because if you're injecting Botox, there'll be no muscle contraction, there'll be disappearance of wrinkles. There'll be disappearance of wrinkles. Okay. Okay, we'll discuss about the curare drugs, ACH inhibitors. Mm, yeah, anticholinesterase inhibitors. Anticholinesterase is basically an enzyme that is degrading the ACH. Okay, is ACH is degraded by acetyl CO acetyl plus choline group. Okay, and this degradation is basically done by an enzyme that is called ACHE enzyme. Okay, ACHE is basically an anticholine esterase. Okay, ase means breakdown. So is basically breaking down your acetylcholine. Okay, so we have many drugs which will break down the acetylcholine. Okay, so ACHE and the drugs that prevent the acetylcholinesterase breakdown is basically called anticholinesterase inhibitors. So in myasthenia gravis, your ACH is not being we need to give this anticholinesterase inhibitors in myasthenia gravis as well, because in myasthenia gravis we need more ACH. So we have to we have to decrease the breakdown of ACH. We have to increase the ACH levels, right? So in myasthenia gravis, we have to give your anticholinesterase inhibitors. We'll look about the anticholinesterase inhibitors, or if you want, we can, yeah, I can discuss the anticholinesterase inhibitors if you want. If you want to know, just stay for 5 minutes or so, and I will. Okay, so these are the drugs which are preventing the breakdown of ACH, preventing breakdown of the ACH. Okay. So in anticholinesterase inhibitors, we have important examples. In pharmacology, I'm giving you an overview of pharmacology as well. Okay. Physostigmine is an important drug. It's an important anticholinesterase inhibitor. Okay. This physostigmine, basically, this physostigmine is a drug of choice for atropine poisoning. So atropine or belladonna poisoning, the belladonna plant is there. Okay, and this physostigmine is a drug of choice. It is also given in your myasthenia gravis, and it is a drug of choice for atropine poisoning as well. Okay, it is basically competitive block. Atropine is basically the competitive blocker of your muscarinic receptors. So to relieve the person from atropine poisoning, we can give your physostigmine. Okay, we can give your physostigmine for this purpose. Okay, another thing, let's know that in myasthenia, where the first muscle to be affected is your ocular muscles. Okay, eye are the first signs of your myasthenia gravis. So ocular muscles are affected first, and at last the limb muscles are affected. Limb muscles are affected the even last. Okay, and the last, and okay, limb muscle last, and again the very last, like the fatal part of the myasthenia gravis, after limb muscle at part, the diaphragm is affected, and when it affects the diaphragm, there will be nothing to save the patient. There will be respiratory paralysis and death. There will be respiratory paralysis. Okay, so basically, first we will do this physostigmine. Okay, we can use your neostigmine and pyridostigmine as well, because there is a drug of choice. The drug of choice for myasthenia, just remember, that the drug of choice is the pyridostigmine. Oral drugs these are, or your neostigmine. This you have to give. Okay, first, these are your anticholinesterase drugs, which will increase the ACH levels. And these drugs will cause bradycardia, that will slow heart. So you can supplement it with atropine as well. Atropine to prevent the bradycardia nature. Okay. And for diagnosis of this, there's a drug called tensilon. Tensilon is a drug which is used for diagnosis of myasthenia gravis. And there's a test in this name which is called the tensilon test. As you're reading physiology, so we are not going to the details of it. Okay, and this Lambert-Eaton syndrome I told is an interesting statement. That this tensilon test is basically used in differentiating between this myasthenia gravis and this Lambert-Eaton. So this LEMS will give a false negative result. The stents will give a false negative result. Okay, so basically this is the thing. And another things you can use for treatment, like first you will give neostigmine. Okay, first you will give neostigmine. If there is improvement, okay, or if there is worsening, we'll see the definitive treatment. Remember the definitive treatment. Oh, I'm running out of charge. The definitive treatment of myasthenia gravis is your immunosuppressants. Understood? Is your immunosuppressants. So just I'm finishing up this class here. I'm running out of charge, and that's all. In today's lecture, we discussed a lot of things, but we could not reach up to muscle contraction. What we just discussed our muscle fiber structure. We discussed the sarcomere and the proteins, and we discussed the synaptic transmission as well as the myasthenia gravis. So that's all in today's class. If you like the class, do like, share, and subscribe. And if you want to ask any doubt, then reach out to me on Instagram at okay. This is my Insta ID. That's all. This is me signing off, and yeah, we'll continue the next part in the next class. And on the next class, we'll read about the neuromuscular junction as well as the muscle contraction. Thank you all, guys. Bye bye. Good night.